Good day, I'm Daryl Scott, and I'm excited to be with you. I wish I could be there in person for this conference, but I'm actually in Guatemala doing a medical mission trip that we do each year with my wife and often with others and other members of our family. Today, I want to talk about this 1P36 Road Trip 2023. I actually talked about this particular title because I gave a similar talk when the conference was in Salt Lake City. That time, I actually did it with my daughter Tiana, who was working in my lab. But I've done kind of a review of this particular talk because I think it's a great way of explaining the differences that we see between different children that have 1p36 deletions. I'm a clinical geneticist, so I work at Baylor College of Medicine, and I have a clinic at Texas Children's Hospital. I see children with 1p36 deletions, but I also see children with a whole bunch of other different genetic disorders. And I spend a lot of my time doing research to identify what are the genes that cause human disease, especially the genes that are on chromosome 1p36. And you'll see that a lot of the things that I'm going to be talking about are things that either I've done in my lab or perhaps other people have done that help us understand a little bit more about 1p36 and the genes that are important. So let's talk a little bit about road trips. Our family went on this epic road trip this summer. We went all the way from Houston, Texas, all the way up into Canada, through Maine, and basically all the Northeast. All in all, we covered about 8,000 miles, which is a lot of trip. But you can have all kinds of different road trips that you might plan somewhere within the continental United States. If you think about it, there's actually an infinite number of different possible road trips that one could take. And every trip is going to have some hazards. There could be rough roads or construction. There's always the risk of having accidents and even running into deer or other wildlife. Some of these hazards are ones that we know and can plan for, others are not. And so it's important to realize that some may be avoided with proper planning or care, but there are some that are just unavoidable. So you're going to perhaps be caught on construction if you want to take a specific road. It's nice to know, of course, what to expect. And so we spend a lot of time planning our trips when we go out as a family so that we know kind of what kind of things we might encounter and are prepared for them. The same is very true for chromosomal deletions, and specifically 1p36 deletions. In this case, there's all these different kinds of chromosomes. They go from chromosome 1, which is the very largest, down to chromosome 21 and 22, which are the smallest. There's an infinite number of possible deletions that can happen. And often I see kids in my clinic where they have a specific deletion, and I tell them, you are perhaps the only person in the world who has a deletion that is exactly that size, or it takes in exactly that part. And so, yes, there's an infinite number of possible deletions, and each deletion can be associated with certain medical problems. Some of those medical problems, again, can be avoided with proper medical care and surveillance. Surveillance meaning that we're testing and looking for problems before they get too bad. Others just can't be avoided. So some things are in place even before a child is born. And so for those particular ones, we know that these things are going to be an issue for those children. It's always, of course, nice to know what to expect, because then you can plan for things and such. And so one of the purposes of a clinical geneticist is to help parents know either what's going to happen for their child or what might be happening or might happen in the future, and then to walk along with them as the child grows and help them get the services that they need. That's what I do as a doctor. We're, of course, interested in this very top tip of chromosome 1, which is the 1p36 region. But again, there's parallels between these road trips and these deletions. One of the things we realize is that everybody's journey is different. Whenever I go to conferences, 1p36 deletion conferences, one of the most common questions I'm asked is, why does my child look so different than other children that have 1p36? Or other people say, well, my child looks like this child, but looks very different than the other. We're going to explain that, or try to. We're also going to talk about what can be expected from different segments of the road or segments of the chromosome and why. But first, let's talk about the road trip part. Why is everybody's journey different? Let's just say we want to take a road trip. This time I'm going to actually mark a road trip out. Let's start from San Francisco, California, let's say, and let's end up all the way on the other side of the country, but we're going to go through some specific spots. We're going to go through Reno, Nevada, Salt Lake City, Cheyenne, Wyoming, Omaha, Nebraska, Iowa City, Cincinnati, Ohio, Pittsburgh, Pennsylvania, and we're finally going to end up in New York City. So we plotted out this particular road trip. And so we know where we're going to go. Now the question is, what are we going to see and what are we going to have to be prepared for? That total road trip is about 3,000 miles, give or take a few. And so it's a long, long trip. When we think about that, most people are not going to drive the entire road trip. In other words, there's some of us who are crazy enough to go crisscross the entire United States. But if most people are on vacation, they may only take a little segment of that road and travel that particular segment, depending on the things that they want to see and do. And so we think about people driving a segment. In the same way, chromosome 1p36 is a pretty large piece of DNA. From the top of the chromosome 1p36 to the bottom of it is about 30 million bases or 30 million letters of DNA. We sometimes write that out as 30 MB, M standing for million, and then B standing for bases. But that's a really, really big piece of DNA. Most people who have a 1p36 deletion do not miss the entire 30 megabases of DNA. Instead, their deletions affect a specific segment of that chromosomal area. And so that's illustrated here. So first we see 1p36 region that's at 30 megabases, but some people just have a little deletion on the top. They may have 99 genes that are affected, or about 2.3 megabases. In this case, if you were to look at it on a road, the comparison would be that this section would be this little tiny section of the road in our map of the United States. And so you can tell that, again, someone who has this piece that's missing is not going to see all of the, instead they're going to see a specific chunk of the United States, and that's corresponding in our metaphor to a specific part of chromosome 1p36. If, on the other hand, you have a bigger deletion, that can take in a lot more genes. In that particular case, we're going to see a little bit different, different medical problems. And so in this particular case, we're going to say, well, what does that correspond to on the map? In this case, if we delete this particular section, it would be similar to driving that portion of the map. And so we're going to see a lot different country, and we're going to perhaps experience different things because the road is longer. We typically know that the longer the deletion, the bigger the deletion is, the more medical problems we run into. That being said, you can have a very small piece of DNA that's missing, and if it contains genes that are super important, you may still have many different medical problems. But we try to figure out what is the segment that's deleted, and then say, what are the problems that might be associated with that? Not all deletions are from the top. Those are called terminal deletions. Other deletions are from the middle. These are called interstitial deletions. They still take out a segment of DNA, but it's not necessarily the most common way that we get 1p36 deletions. In this particular case, this might be 3.5 megabases. That might correspond to this piece missing, and then this particular part of the road in the United States. And so you can see that every child can have a different deletion, may have different genes that are affected. We don't see the same deletion over and over again in different children with 1p36 from different families. So that's a little bit of why everybody's journey is a bit different. If we think about it again in terms of the road trip, if you start out in the West, we're going to see certain type of land. You have mountains, etc. If you hit the Midwest, it's going to be very, very different. And once you hit New York City, it's going to be, again, very, very different. Each one of these areas can have its own problems. That's why we expect different things. So if you're out in the West, you might have dust storms. That's something that we're used to, and we know kind of how to deal with that. If you're going to be in the Midwest, you're going to see farm equipment, and you may end up having to stick behind some piece of farm equipment for a long way, especially on these dirt roads. In New York, you may have taxi cabs and just a lot of traffic. Again, each section of the country has its own issues. And if you're driving through that, you're going to expect to hit different problems. In much the same way, there's different parts of chromosome 1p36, and those pieces are associated with potentially different problems. Now, not only are we gonna run into a dust storm or a piece of farm equipment or even the taxis that are crazy, but these are all possibilities. In the same way, I'm going to show you segments. I'm going to tell you what are possible causes or what are possible things that you might see if that segment is deleted. So the first question though is, how do I tell what segment is affected in my particular child? Nowadays, almost all children with 1p36 deletions are diagnosed using the same technique. That technique is called chromosome microarray analysis, or sometimes we say CMA, chromosome microarray analysis for short. This is the actual test that's done in order to be able to detect one of these deletions. How this is typically done is we take either a blood sample, in which case we're interested in the white blood cells, and we're going to extract DNA from those white blood cells because our red cell DNA doesn't have our red cells don't have a lot of DNA. More and more these days, that can also be done using just a swab, and we'll take some of the cells around the cheek area and gum area and use those to extract DNA. This particular test can be done with any tissue that contains DNA. And so it could be done with a lot of things, but blood and saliva are the most common these days. We're then going to take our sample of DNA and we're going to put it on a microarray. A microarray really looks like kind of like a glass slide, like you might remember from when you were in science class, you looked under the microscope and you had a little glass slide. The size of those are almost exactly the same as those glass slides, but these slides are imprinted with thousands of little probes. Each one are on a particular circle of that slide. And I'm going to show you that a little bit more, but literally you can hold one of these microarrays between your fingers. It's just a piece of glass. We have chromosome here, 1p36. Again, the idea is that you have probes all the way down that. And in each probe, we're looking at a unique part of DNA that is, again, unique to that section of the chromosome 1p36. And then we're basically going to ask with this test, how many copies of DNA do we have in our cells from each one of those probe areas? After we prepare everything, we use a laser scanner to scan these microarrays. This laser scanner costs way more than my house. Not everybody has a laser scanner. These days, most of the laser scanners are going to be in laboratories that do clinical testing because most of the tests that are done this way are done in the clinic. However, we sometimes still do these on a research basis. I started using microarrays when they first came out, and we were lucky enough to have a laser scanner that was located at Baylor College of Medicine that we could use. But again, most of these are now going to be in clinical laboratories. So what does a microarray look like when we get it all processed? So this is a picture of a microarray result. In other words, this is the raw data from a microarray. This, as you can see, it looks kind of like a box because it's on that slide. But if you blow it up, you actually can see that each little dot is actually a little circle. That circle is a DNA probe that's actually printed onto the slide using a giant printer, like an inkjet printer. But instead of squirting ink onto a piece of paper, it actually puts down bases of DNA onto the slide in exactly a specific circle. That printer is about the size of, well, a good chunk of my house. How's that? One level. It's a very, very large printer that prints these microarrays, but it does so very exactly, and it literally builds DNA one base at a time. Then we take the patient's DNA and we hybridize it or stick it to that DNA, and it can tell us how many copies of DNA are there. You probably already noticed that there's different colors. Some of the circles, for example, are green, others are yellow, and others are red. This is because the computer scanner, the laser scanner that we use, actually colors them in different colors for us, depending on how much DNA is stuck to that area. And we use a patient sample and a control sample, and it tells us the ratio of the DNA between those two samples. All you need to know is, again, we can tell whether a single area of the DNA has one copy of the DNA, two copies of the DNA, which would be normal. Most of us have a copy from mom and a copy from dad, or three copies of the DNA. 1 corresponds to a deletion, 2 is normal, and 3 means a duplication or an extra copy of DNA. Most times when we look at the slides, we can see that they mostly look yellow because most of the DNA is normal. But sometimes you can actually look at the slide, and if there's a big enough deletion, it will look green. Or if there's a big enough duplication, it will look red. That's because all these little circles are turning red, and your eye can see the difference between some percentage of the probes that are red or green versus one that's all yellow. This is still not usable. In other words, even if you could look at this, the slide is not meant or not made so that each chromosome follows one after another. Instead, it's mixed up. So each probe is in a different place. The reason for that is you don't want one piece of the slide to be hurt or not to be correctly processed, and that might affect your result. So they have a random pattern. But because of that random pattern, you then have to use a computer to put all the probes back in order. When they're all put back in order, we get a readout that looks something kind of like this. As you can see, we can see all the different chromosomes and each section of the chromosome, but we're going to actually concentrate on a part of the DNA, which is shown over here. So if you say this is chromosome 1, this is chromosome 1. In this particular case, we're looking at a deletion that's kind of at the bottom end of chromosome 1. This is 1p36 up here. But this is a blow-up of that region, and you can see that each one of these dots represents a probe. In this case, black is normal, green is a deletion, and these little red ones, if they were all clumped together, would form a duplication. There's some variation in each one of these probes, and some probes work better than others. So a single dot on one side or the other is not important. We're looking for a group of dots all together that form a deletion or a duplication. We can then blow that up. And when we blow it up, you can actually see that we can put lines on either side and say, that's exactly where the deletion occurs. Then we can say, well, what are the genes in that area? In this case, all the genes are like written out here in gray. And so I can actually use this particular picture to say what genes are involved in this specific deletion and where the deletion begins and where the deletion ends. That all is transferred into a report. This is a report of a child that does have a 1p36 deletion. In this particular case, the part that's most important are these little boxes right here that tell us the information. On this report, we have the place where the DNA is deleted, and they give the chromosome numbers. And so chromosomes are chromosome 1, P, 3, 6. That's what we talk about, that whole big region. But then there's sub-regions like 1 and 1. And so we read this as 1p36.11. And that for a geneticist means a very specific piece of the DNA. This deletion goes from that area to 1p35.2, which is the section of the DNA that's just below chromosome 1p36. So this is an unusual deletion in that it comes right kind of at the very end of chromosome 1p36. This particular lab gives us a minimum interval, and you can see that's represented by numbers. This is actually how many letters you count in from the top of the chromosome, and it says the deletion perhaps starts from here and goes to here. The problem is when we do these arrays, not every specific piece of DNA or every letter is caught. So there's a little distance between each probe. And so you can imagine that sometimes we know that this piece is deleted, but there's some uncertainty. So we give you a minimum and a maximum region. Other labs do it a little bit different and only give you one set. And they'll just say, we know that this particular part is deleted. Each lab is a little bit different, but we're looking for this type of information. You can see that this deletion is between 3.4 and 3.5 megabases in size. So kind of summarizing this, I would take this section and we can summarize it by saying this is a loss or a deletion. This is the chromosomal area, and this is the maximum interval, and the size is 3.1 megabases. This is what I need as a starting point as a geneticist. Without this information, I can't really tell you very much about your child's 1p36 deletion. because again, each child can have a different piece of 1p36 that's missing. I have to have this data and then I can begin. So let's talk a little bit more about these things. Some of these deletions, again, on the back end, this would be the deletion that we just looked at. This is an unusual deletion far, far away from San Francisco. It's actually up into the New York area and a little bit farther. That's the deletion we just looked at. So let's talk about a few things on chromosome 1p36. Let's talk a little bit about critical regions or critical genes and critical regions. What they do, I'm going to put that in, well, I'm going to put the gene or the region in red, what they do in black, what problems may be encountered because if that gene's deleted, and then what we can do to avoid or minimize issues. So let's start. One of the first genes that we think about in a chromosome 1p36 deletion is GNB1. It's one of the very first genes on the road that are important. It plays an important role in transferring signals from outside of a cell to the inside of a cell. Children that have this deleted can have a number of medical problems. They can include global developmental delay, so that can be problems with how they walk, how they use their fine motor skills, and also how they develop language, intellectual disability, vision problems, seizures, poor growth, and it may also play a role in developing cleft palate. Once again, some things are very likely, others less so. So almost all the children that have this gene deleted have global developmental delay, and most have intellectual disability, but there may be variations in how severe that is. Many of them will have seizures, but some do not. Many of them have poor growth, but some do not. And many have cleft palate, but some do not. Why is that? Well, you have to remember that every gene has two copies. And so in this particular case, we're going to delete one copy, but there's one copy that's still working in most children. That one copy, if it's working really, really well, might decrease the amount of issues we find when the other one's deleted. If, on the other hand, this gene also has some damage to it, the problems could actually be worse for a child that has this deleted. And so there's great variation we see even within the segments. If two children have the same segment that's missing, they may still have differences in their medical problems. But what do we do about these things? For global developmental delay, one of the most important things we can do is therapy. Physical therapy, occupational therapy, and speech. Sometimes we don't think this is as being like a medication, but often this is the best thing we can do for children. Physical therapy, occupational therapy, speech therapy, these can make a big difference. And so it's important that we have these kind of things available for our children that have 1p36 deletions. It's important that they have a balance. And we realize that you can't spend your entire life in therapy. We totally understand that. But having some of these therapies is a very helpful, and certainly kids can make a lot more progress if that's available. Intellectual disability usually is an issue that we face. We think about that in children when they're starting to be in school. That's mostly going to be talking about accommodations or extra help in school. As clinical geneticists, we try to help parents to negotiate for accommodations in the school system. We recognize that different parts of even a city as big as Houston will have different levels of like accommodations that can be done in different schools. We wish that all of them could do the maximum accommodations, but we realize that's not really practical. But we do write letters indicating what children need as far as help goes. And especially if we need extra help in knowing what a child needs, we can send them to a developmental pediatrician who can then do a very thorough analysis and then provide recommendations to a school system. This is something you might want to consider for your child if you're having difficulty knowing how to maximize what they're doing in school. Again, that's a developmental pediatrician. They specialize in trying to do the tests that help schools to know what to do for children that need special accommodations. Vision problems, we always like all children that have any kind of delays to have a yearly vision exam. For vision problems, we're going to be mostly wearing glasses or some other kind of correction, but some kids will also need surgery. Seizures are typically treated with medications that help block those. Not all children that have seizures are going to have them forever. And so it's important to have a neurologist that both helps to determine what the medication should be and also help to determine when the medication may no longer be needed. Every medication has some side effects, and so we want to minimize the amount of medications while at the same time controlling seizures. Poor growth, sometimes having nutritional support or even growth hormone can help in maintaining normal growth. And so that can take many different, can be done in many different ways, but often our children are followed by gastroenterologists who help us, and they help determine how we can help our kids to have optimal nutrition. Cleft palate, mostly that's feeding support when they're young, and then ultimately these kids will go to surgery to have their cleft palates repaired. All of these things can happen as part of having just one gene deleted, that's GNB1. And a lot of the children that have terminal or deletions that start from the top will have this gene deleted. GABRD is a chloride channel that's triggered by an inhibitory neurotransmitter. In other words, this has an important role to do in brain. Because it has a role to do in brain, the thing that we think about mostly in children with this particular gene deleted is seizures. Not all will have seizures, but again, it increases the risk for seizures when this is deleted. And how do we treat that? We treat that with medication. PRDM16 is probably one of the most important genes. If there's a gene that I think about more than any other, it's probably this one. This gene binds DNA and changes how genes are turned on or off. It is very important because it can cause cardiomyopathy. So if we have PRDM16 deleted, children can have either at birth or develop a problem with their heart muscle itself. And the muscle does not contract like it should. As a result, they can have heart failure. It's really important that we monitor for this. Again, some children are born with problems with this, others develop it. And so one of the things we suggest is that anybody who has this gene deleted should have an echocardiogram. Currently, we're indicating that we should have echocardiograms at least once every 3 years for any child that has this gene deleted. In other words, if the first echocardiogram does not find cardiomyopathy, they should still go back in 3 years and be re-screened for evidence of cardiomyopathy. And we also think about this anytime a child has difficulty with being extra tired or having other issues that would be similar to what we might see in a child that has heart failure. So they're having a harder time doing the activities and such that they were doing before. And there's no other reason we can find. We think about this, especially again in children that have PRDM16 deleted. If you can identify the cardiomyopathy, often we can manage that with medications, but it can be so severe that some children have to go to cardiac transplant. So this is a very serious gene that we really think about, and it's important that any child that has this deleted be monitored carefully. We sometimes think about not only just genes, but also regions. So sometimes you may hear about this thing called the distal critical region. Children that have deletions that involve this region have kind of that what we consider that classic 1p36 deletion syndrome. Other things we haven't talked about that these children can have include congenital heart defects, hearing loss, and hypothyroidism. But they can also have all the other issues that we've mentioned before. This distal critical region takes in a certain part of that map that starts from the very tip of the chromosome and goes in a little bit. That's shown here, down here on the bottom. For congenital heart defects, we think about monitoring, or sometimes the kids will need surgery. For hearing loss, we think about amplification, so like having a hearing aid. Hypothyroidism is low thyroid levels. That's really easily treated, but it does require children to be screened to see if they are hypothyroid and then receive medication. Typically, it's a medication that's taken every day. It's Synthroid, which is basically a thyroid hormone. But it doesn't stick around very long, so it needs to be administered or taken as a pill or a liquid every day. There's also another region called the proximal critical region. It has all the same problems that are associated with a classic or distal 1p36 region. It takes in this particular section of the DNA. So you can see there's a little bit of a break between the distal or like farther away from the center of the chromosome, to the proximal, closer towards the center of the chromosome. But again, children that have this particular region deleted have all the same medical problems associated with classic 1p36 deletion syndrome, but they have different facial features. You may be able to see that in the difference between these two children. So some of the things that we think about are the eyebrows that are very, very straight, kind of deep set eyes. Certainly this child who has a proximal deletion does not have those curved, those straight eyebrows. It just has a different look of his face. That's what we're thinking about when we think about different genes. So both these regions are non-overlapping. They cause different or similar medical problems, but they're caused by deletions of different genes. And that's why the facial features are different, because the genes that are different that are actually missing in these children. Again, this particular part of the DNA. In that proximal critical region, there's a gene called RERE. We think it's one of the most important genes in that proximal critical region. It regulates how genes are turned on and off. And just by deleting even that one gene, RERE, you can get almost all the features that are associated with a deletion of the proximal critical region. So we think this gene is very important and is one of the genes that we work on the most in our laboratory. These distal and proximal critical regions, most of the children that have 1p36 deletion syndrome will be deleted for one of these regions. Whenever we see those, we know more about those children than we do about children who might have deletions along the other parts of chromosome 1p36. So these are the ones we kind of think about as doctors when we think about 1p36 deletion syndrome. But your child, again, may be different because they have a different segment affected. Let's go way down the list into an area that's outside those critical regions. There's another gene called KDM1. It, again, is important in how it turns genes off. This gene, if it's deleted, can cause developmental delay and cleft palate. But again, it's far away from all the other genes. GRHL3, it also turns genes on and off, and by doing so, stimulates cell migration. Cell migration is really important in how we develop certain organs. And it's also very important in how we develop our palate. So when you miss this particular gene, kids get cleft palate. Or at least are at risk for having cleft palate. ARID1A or ARID1A, it's again a gene that turns genes on by making DNA accessible. So when we turn a gene on, what happens is the DNA is all wound and can be kind of very tight. And the things that turn on a gene and actually make the protein that gene makes can't get to that DNA. So they can't get to the plan or the gene that's required to make that protein. This particular gene opens up the DNA and allows access to that area. As you can see, a lot of the genes that we've talked about actually do that same kind of thing. They turn other genes off or on. They're called transcription factors. So ARID1A is a type of transcription factor, but it opens up the DNA. Children that have this gene deleted can have developmental delay and intellectual disability. It also plays an important role in heart development, and they can also have short stature. AHDC1, again, a gene that turns genes on and off, developmental delay, intellectual disability, growth problems. All these genes are genes that are of importance. You could ask, how many genes are important on chromosome 1p36? Actually, that's something that we're still working on. So we're still learning what a lot of the genes do. How do we determine if a gene is important? And also, how do we determine what they do? Let me kind of run through with you how we do this gene discovery process. One of the important ways that we identify genes that are important is actually by using normal patients. Normal patients means normal individuals. They're individuals who are living, often have families, kids, have a job, etc. At the same time, every normal person is not exactly normal. I consider myself a relatively normal person, but I've had 5 surgeries during my life, right? And so I have medical problems just like you do. But when we say normal individuals, we're basically saying don't have major medical problems. that would have stopped them from, let's say, having a family, having a job, etc. So there's a database that has 141 plus thousand individuals who are normal, and they've done DNA tests on these individuals. At first, when I heard about this database, I thought, what a waste of money. Like, who cares about normal people? But as it turns out, it really helps us a lot, because one of the things you can do is to look at a gene and say, well, based on the size of this gene, we assume that by chance, just by chance, this gene will be disrupted or hurt in a specific number of individuals. In other words, we can predict how many loss of function variants we'll see in a specific gene. So let's take a gene that's of reasonable size. We may say that based on the size of the gene, we would predict that in this group of individuals, 45 would have a mutation in that gene that would make it not work. We then go back to the database and say, well, how many people actually have that type of change? And let's say it's 42. Those are almost exactly the same. One of the ways you can think about that is to say, well, okay, that means that having a change in that gene or having that gene not function does not take a person out of the normal population. In other words, you can have a change in that gene and it doesn't matter as long as it's at least only one copy is affected. So since these are very similar, those two numbers, we consider that this is not a bad gene. And we give it a score. In this case, this is the probability of loss of function intolerance, or PLI score. It goes between 0 and 1. Because these numbers are very similar, this would get a PLI score of 0. I'm not particularly interested in these genes. These are genes that are not likely to cause a problem if one copy is deleted. So when I see a PLI score 0 gene on chromosome 1p36, I'm assuming that that's not causing much of a problem for my patient. In contrast, if you have a gene where you expect 45 changes, but in the normal population, you only see 5, that's a big difference. Another way of thinking about that is a change in this gene has caused 40 individuals to be removed from the normal population. This kind of gene is bad when you delete only one copy. And it gets a PLI score that's not 0, a PLI score of, in this case, 1. So this has my complete attention. When I see a 1p36 gene with a PLI score of 1, I'm assuming that that gene does something very important and that can cause medical problems for my patient. Let's think about PRDM16. Remember, that's the gene that causes cardiomyopathy. So what does it look like when we put it through this particular pattern? In this case, PRDM16 was thought to have about 49 changes that were expected based on its size, but they found only 4 individuals that carried a PRDM loss of function variant. This is a bad gene, has a PLI score of 1. That makes sense? RERE is another bad gene. This is the one that I work on, and it can cause almost all the medical problems by itself that we see in 1p36 deletion. It was supposed to have 65 changes based on its size, but we found only 3. Again, this is a PLI score 1 gene. It is a bad gene. We're worried about this if a child's deleted for it. Let's talk about another gene that we very recently have been working on. This is a gene called SPEN, or SPEN. It kind of is almost right in the very middle of things as far as our map goes. SPEN is an important gene in that it turns other genes on and off, just like many of the genes that we've been talking about. But how do we know if deleting one copy of SPEN is enough to cause problems? Well, this is the actual database that we go to get our information. So we type in SPEN into this database, and we're interested in this little box of information over here. It gives us our PLI score. So in this case, this is a very big gene, so we thought that 129 people would probably have a change in this gene that would make it not work, but we found only 4. That difference is really, really large. So this has a PLI score of 1. It is a very bad gene. Then we say, well, what does that gene do? In other words, even though we know it's a bad gene that might cause disease, we don't know what disease it causes. It could cause a heart defect. It could cause some problems with brain development. So what we didn't do is we go to the thing called Gene Matcher. Gene Matcher is a great place for people who hunt genes like me, gene hunters. We're enrolled in Gene Matcher, so I log in, and basically what it allows me to do is to say, I have an interest in a gene called SPEN. I put that into the database and it sends me an email that gives me the names and emails of all the other people who are interested in SPEN. I then send out an email that says, hey, I have a patient that has a deletion of SPEN. They have these kind of medical problems. What do your patients have? Then we talk amongst ourselves. And if we see a pattern forming, then we know that that's what that gene does. This is exactly what we did for SPEN. Specifically, we got 34 individuals that had damaging variants in SPEN. We then looked at what the medical problems each of them had and compared them. Based on this pattern, we then can say, this is what SPEN does when you delete one copy of it. We then write a report. So this is the actual report that we wrote together. You can see my name's on it, and also some of our collaborators' names are on this. And we say that this particular gene is important for developmental delay, intellectual disability. It can cause autism spectrum disorder. It can cause anxiety, aggressive behavior, attention deficit disorder, hypotonia, which means low muscle tone, brain and spine anomalies, congenital heart defects, high narrow palate, facial features that are unusual, and can also play an important role in obesity, especially in females that have this gene deleted. This gene is important in how the X chromosome in females, females have two X chromosomes. One of those has to be condensed so that only one of them is turned on in each cell. This gene plays an important role in that. So some of the findings are actually specific to females. And we found a difference in the obesity, mostly in our female patients that have SPEN deleted. This helps us give an understanding of what this gene does. Even though we've done all this work, we're still finding out more about SPEN. One of the ways we do that is we also use mouse models. So in our laboratory, so I'm sitting in my office, my laboratory is just down the hall. We have SPEN deletion mice. We know that SPEN causes problems with brain anomalies and heart defects in mice. That was already known when the gene was discovered to be important for humans. But since that time, we've used those mice to learn more about how does SPEN work and how does SPEN cause heart defects and brain defects in people. One of the things we've also learned is that individuals that have SPEN deleted and mice that have SPEN deleted have cleft palate. This is something we did not realize to begin with, but there were a few individuals that we found that had cleft palate, but all of our mice, when we delete SPEN, have cleft palate. And so this is a cleft palate gene as well. So we sometimes use humans to help us understand what a gene does, and sometimes we use mouse models to understand what a gene does. But most times, to tell you the truth, we use both. We get information from humans, we check it in mice. We see what the mice have, and we check it in humans. By doing this, we get able to understand more and more about how things work. I told you that SPEN kind of sits in the middle. Well, one thing I did not explain is that RERE, which is over here, is another gene that we work on. The reason why we work on both of these genes in our lab is because RERE actually has a role in turning on SPEN. So in other words, some genes, like I said, turn other genes on and off. Well, they actually can interact with each other. And so SPEN can cause brain, heart, and palate problems. RERE can cause brain, heart, and palate problems. And so you can imagine that perhaps the reason for this is because RERE's job is to turn on SPEN. In other words, they work in a pathway together. And by disrupting anywhere along that pathway, we end up with brain, heart, and palate problems. This also can help us to understand how we might help treat a child that has one of these genes deleted, because we could potentially turn on SPEN or turn up RERE. And if we could, we might be able to make some of these medical problems better. That's the idea. There's, of course, some things that may be too late for us to do even after a child's born. So, for example, Brain abnormalities are the brain's going to form very early, even before birth. Heart also forms before birth, and palate forms before birth. And so this is still a real challenge. And so many people say, well, how will we ever help? Or is there any way that we can help the children that have these deletions? The answer is some of these genes also play an important role after birth, and it might be possible to do some kind of therapy to help. But for now, most of the 1p36 deletions that we see cannot be helped through that kind of gene therapy. Many of them contain many different genes, and it's quite challenging. And again, not all those genes actually have an important role after birth. Many of them have an important role very early on in development. But anyway, we are interested in learning how one gene can affect another. One of the ways we can think about this as being useful is if we know that two genes are in a pathway. Well, we would expect that if you delete both RE and SPEN, which happens in some children, the risk of all these problems is going to go up because now you've actually hit two different genes that are important in this pathway. And so by learning that, we can also understand a little bit about why some children have more medical problems than others, because how the genes interact within a segment of DNA can also be important. One of the common things we also are asked is, well, if my child has a specific deletion that could cause, let's say, seizures, but they're seizure-free, why is that? You have to remember that we have 20,000 genes, and those genes interact together. And so your child is missing perhaps one copy of several genes, but all those other genes are also making a difference, turning things on, turning things off. Our environment can also make a difference. And so that's why individuals, even that have just the same change in a gene, can be very, very different. Sometimes we see differences even within the same family that have the same gene or segment of DNA deleted. This is the kind of variation that we expect because our systems are very complex, and each gene has some unique features. Some are working a little bit better, some perhaps a little bit worse. And because of that, we have variations in the symptoms that we see. One of the things that also parents ask is, how can I stimulate interest in my child's section of chromosome 1p36? Many of us are working on genes that are very, very important, like RERE or SPEN. We're one group that works on that, but other groups around the world work on that as well with us. And so there's some areas of the chromosome 1p36 that are highly investigated, but your child may have a section of chromosome 1p36 that's unusual, or perhaps especially small. We're interested in those particular children because they can give us clues to areas of the chromosome 1p36 that we don't know a lot about. So how can you develop interest in a specific region of the DNA? Well, one of the ways you can do that is to join our research efforts. What we typically ask for is for children who want to, or parents and children who want to join our efforts. We ask that you fill out a consent form that can be sent by email and filled out. We then want a copy of the lab report that shows the specific area of the DNA that's been deleted in your child. And then, especially again, if it's small or in an unusual place, we can then use that to learn about the genes that are involved in that region. I'm not at the conference, sadly, but I can be reached on my email. My email is dscott, stands for Daryl Scott, at BCM, which stands for Baylor College of Medicine.edu. If you want to email me and say, we have interest in joining your research study, we would be happy to visit with you, learn more about your child, get a copy of the lab report, sign consent forms, and then you can be part of our database. That particular database, again, is something we use over and over again in papers to determine what genes are important in 1p36. My last couple of slides are just to thank people. I've had many, many people who've worked on different genes in our laboratory, but probably the most important ones that work on kind of identifying which genes are important are individuals like Bumjun Kim, who does a lot of our mouse work, Andres Hernandez, who does our mouse work and also helps us out with the human studies. We've had several students in our lab. They include Hatisha Zaviri, Valerie Jordan, and even my daughter, Tiana Scott, actually worked in our lab for a while. And because she had gone to so many 1P36 conferences with us as a family, she said, Dad, I want to work on a 1P36 project too. And so she's been important in a lot of the projects we've had that talk about cleft palate and other issues in children with 1P36. We also have we need a lot of money to be able to do this research. Most of our money comes through various governmental organizations. I work at Baylor College of Medicine. I see children at Texas Children's Hospital. But we've received funding from the March of Dimes, the American Heart Association, and especially the National Institutes of Health through the National Institutes for Child Health and Human Development. We appreciate that kind of support. We also are very, very grateful for all the families that have joined our studies. We recognize that this is a challenge. We wish that your family did not have this challenge and that your child was able to live without all the difficulties they have. At the same time, being a parent of 7 kids, I realize that every single child has something. And it's just that we want to understand what your child has so that we can better know how we can help that child and help other people who have similar deletions to that child. But thank you for your help and support throughout the years. I wish I could take questions, but if you really do have a burning question or something you want to ask, I encourage you to email me. Again, it's dscott@bcm.edu. Pretty simple. I get emails here and there from all over the world with individuals who have children with 1P36 deletions, and I try my best to answer their questions. Of course, it's always important to have a local geneticist, and I wouldn't suggest that anybody needs to actually come see me. We can do very well for almost anybody in the United States as long as you have a decent geneticist and a pediatrician who wants to help. We can help you do whatever you need to do right where you are. Thanks again for taking time with me, and I hope you have a great conference.